Good afternoon, students, and welcome to practice problem session for week 5. It's 3 p.m. Let's wait for 5 more minutes and we'll admit other students trying to join this call. Once we reach 4 or 5 students, we'll start the presentation. Meanwhile, the instructions would be same. You have to turn off your mic and video chat during the session. If you have any doubts or questions, you can raise your hand and speak up. But all communication with me will be via the chat box. So you mention your answers in the chat box. If you have some small doubts, you can raise your hand and speak up. Let's wait for 5 more minutes and admit some more students, and then we'll start at 3:05. I'll start presenting my screen, and today we'll be solving practice problem corresponding to lecture week 5, where Professor Subramaniam has covered cell communication, part 1 and part 2. As you can see, it's already 3:05, and I'm going to start the NPTEL problem solving session right away. So let me share my screen, and the format would be same for those who have attended the earlier classes and those who have not. I'll lay down the general instructions. So the way we'll be proceeding is with solving 10 questions corresponding to the current week lecture, which is lecture 5 for this week, and consecutively we'll move on to lecture 6, 7, 8, 9, 10, 11, 12 till the end of the course, right? And these questions would be from those corresponding lectures and will be similar to kind of your quizzes and assignments on which you will be graded. So we'll be solving problem set 5 today, and these problem will help you solve the actual assignments and the quizzes that you get, and also help your understanding in the final exam. I think not many people have joined. It's only Ajay that has joined. Hello Ajay. Anyway, we'll proceed with the class. For those who have missed this session, they can avail this recording. So it won't stop me from taking this class. Ajay, you can mute your mic so that, yeah, great. So that I'll start presenting. And meanwhile, if somebody asks to join this call, I'll keep admitting them. So let me present my screen quickly. I hope you can all see my slides, and can also see the slides changing. The network on my end might be a little slower, but let's deal with it. Great. So once again, I welcome you all to practice problem set week 5. So in the week 5 series of lecture, Professor Subramaniam has covered concepts of cell-cell communication. So we'll be dealing with questions related to that. And again, we are in the course Introduction to Developmental Biology, and this is practice assignment for week 5. The course instructor is Professor Subramaniam, and my name is Ashutosh. I'm a PhD scholar at IIT Kanpur, who will be the TA for this course. So the first question that we are going to solve from practice session week 5: which of the following can directly or indirectly affect the surface tension of a cell? This is a question about surface tension. Which of the following can directly or indirectly affect the surface tension of a cell? Option A, type of cadherin molecule. Option B, amount of cadherin. Option C, actin polymerization. Option D, Rho GTPase activity. Option E, phosphorylation of catenins. Or option F, all of the above. So here is the question. Yeah, yes, Ajay, if I reveal the answer, you will know that you are exactly correct. All of the above is the correct answer. So all these phenomenon can directly or indirectly affect the surface tension of a cell. So the type of cadherin molecule, amount of cadherins that are present on a cell, actin polymerization, which is a part of the cytoskeleton, Rho GTPase activity, which is an important enzyme involved in polymerization of microtubules and actin, phosphorylation of catenins. So we will learn about these concepts shortly. So first let's learn about cadherins. Cadherins are transmembrane proteins that mediate cell-cell adhesion in animals. So any two animal cell can have these transmembrane proteins. Transmembrane proteins, when I say, I mean these proteins have passed the plasma membrane one time. They have an extracellular domain, they have an intracellular domain. If you see here an example of the cadherin protein, this is the transmembrane domain. The part that is outside the plasma membrane is the extracellular domain, and the part of the protein that is inside the plasma membrane is the intracellular domain. So cadherins are all these transmembrane proteins which mediate cell-cell adhesion. So they are very important in cell-cell adhesion property. By regulating contact formation and stability, cadherins play a crucial role in tissue morphogenesis and homeostasis. So where is that cadherin required the most? In maintaining the tissue morphogenicity and homeostasis. So when we have cells arranged in a layer, those cells in that layer are adhering to each other and to the layer via these cadherin interactions. That's how we have sheets of layers called epithelial tissue, and all these tissue organization that we have is due to cadherins. One of the most important factors is cadherins. And cadherins also have another property of homotypic interactions, where we can see that cadherin molecules from same subtype have similar extracellular domains which are responsible for their adhesive properties. So when we have two similar types of cadherin, so there are many types of cadherin, prominently E cadherin, N cadherin. So if we have two molecules of E cadherin of the same type of cadherin, then they have similar kind of extracellular domains. Now similar kind of extracellular domains will form a strong adhesive interaction. So that is the property of cadherin. Similar type of cadherins bind with homotypic interactions, and also when cells expressing the same type of cadherin come into contact, the cadherin molecules on their subsurfaces bind each other in a calcium dependent manner, forming what is known as adherence junction. So here I have mentioned a typical junction which occurs in epithelial cells, which is an adherence junction, and the adherence junction harbors all these cadherin interactions in a calcium dependent manner. So E cadherin will bind to another molecule of E cadherin in the presence of calcium and attach these cells together and to the membrane. This is known as adherence junction. So there are many components of adherence junction. So here we can see that the type of cadherin can play a role. And if we focus on this diagram, the intracellular domain of cadherin also has binding sites for many other proteins like catenins, beta catenin and alpha catenin, and other adaptor proteins P122, which can attach this cadherin interaction or complex to the actin cytoskeleton that we see here in the purple, or via P120 it can go and modulate microtubule dynamics. So this is how a cadherin interaction at the extracellular cell membrane is transferred into the intracellular cytoskeleton of actin and microtubules. Now we focus on the actin cytoskeleton and actin polymerization and also the catenins. Actin filaments can exert mechanical forces on cell membrane leading to changes in cell shape and membrane tension. So how can actin actually affect the surface tension in a cell? So actin has the property to polymerize and depolymerize in a constant way. It is polymerizing, some ends are depolymerizing, and these polymerization and depolymerization leads to growth of certain microfilaments which are made up of actins. So here is the process of actin filament assembly. We have G actin. G actin refers to the subunit of actin filament, globular actin for G actin. In presence of ATP, when ATP is bound to G actin, then G actin forms stable ditrimers, stable actin oligomers, and then filament assembly is mediated. And then when ADP is bound to actin, then disassembly is mediated. So this is how an actin forms from a subunit of G actin bound to ATP gets activated, and then ATP bound actin forms a filamentous structure called F actin. And this is how a microfilament can form. And when this microfilament is polymerizing and polymerizing, it can hit the cell membrane. It can exert the mechanical forces on cell membrane, leading to changes in cell shape, morphology, and membrane tension. So when actin polymerization, so if you imagine in a cell full of cytoplasm, which is liquid, we have these polymers of actin, of G actin called F actin, which are growing towards one end and which are depolymerizing from the other end. So the end which is growing, that will eventually hit the cytoplasmic membrane, which is the plasma membrane. So when it hits that, it can push against the membrane, cause it to protrude forward or outward, forming extensions like lamellopodia and filopodia. So these are characteristics of actin cytoskeleton structures. Lamellopodia or feet-like or webbed feet-like structures, where actin polymers are growing towards one end. You imagine that they are pushing the cell membrane outwards and forming a podia. Podia means feet-like structures. And filopodia is fine projections. So all these structures can form, and in this way actin can lead to change in cell shape and also surface membrane tension. Now we come to catenins. Catenins are the proteins which bridges actin filaments and cadherins in a cell. So as we saw from the previous slide, these are the two catenins here, beta catenin, alpha catenin. So what they do, they can link these cadherin adhesion forces to the internal cytoskeleton of actin filaments. This linkage transmits the force among the cells, the ECM and cytoskeleton. So this linkage is essential for transmitting the force. So how is force generated? A cell is binding to its neighbor or to the membrane via this adherence junction where we have cadherins, and then we have actin filaments which is forming the cytoskeleton of the cell, and we have certain proteins called catenins which link the adherence junction with the cytoskeleton. So that's why we have the correct answer as all of the above, where the question was which of these following can directly or indirectly affect surface tension in cell. So we saw that type of cadherin molecule will play a role, because same type of homotypic interactions that we studied about. Amount of cadherin, if there is more amount of cadherin, then we can have more homotypic interactions and more adhesiveness, more adherence junction. Actin polymerization we just saw. Rho GTPase plays a role in actin polymerization and also microtubule dynamics, phosphorylation of catenins. So here is the third player, Rho GTPase, which I was talking about. Rho GTPases are a family of small GTP binding proteins that act as molecular switches, cycling between an active GTP bound state and an inactive GDP bound state. So Rho GTPases are protein which bind to GDP and GTP. So when they are bound to GTP, they are active, and when they are bound to GDP, they are inactive. So here we see these microtubules. These GTPases are involved in microtubule polymerization dynamics, which can also lead to change in surface tension in a cell. So if we see here, microtubule is made up of subunits of a heterodimer called alpha beta subunit. So these are tubulin proteins. That's why the name microtubule. It's easy for you to remember. So these tubulin dimer, heterodimer of alpha tubulin and a beta tubulin, when bound with GDP is inactive, but when bound with GTP is active. So the similar way actin is bound with ATP, then it is active, and with ADP it's inactive. And G actin is a subunit of microfilament, while alpha beta tubulin dimer is a subunit of a microtubule. Then these GTPases can actually change the state of GDP bound to the dimer or GTP bound to the dimer, thereby providing the subunits for microtubule polymerization at one end and depolymerization at the other end. So GTP hydrolysis will form the minus end where we have polymerization, while GTP where we have depolymerization, while at the plus end GTP bound tubulin units can come and polymerize to form the growing end of microtubule. And these microtubules also function in forming the tracts on which different motor protein transport cargoes. Microtubules are also very important during cell division, where we have separation of chromosomes. via microtubule pooling. So microtubule also generate lot of force. So here we saw that rho GTPase, phosphorylation of catenins, the type of cadherin, amount of cadherin all play an important role. All play directly or indirectly a role in changing the surface tension of the cell. I hope it is clear to the students. I see a few more students have joined. Just let me know in the chat if it's clear. Then we are already moving to the second question. I can see Ajay and Simontini. Sakshi has also joined. I make sure that nobody has missed the first question, so I reiterated it while you guys were joining. Is the explanation clear for the first question, that all these processes can change the surface tension of the cell? Yes, good. So moving on to the second question, it's already on your screens. So if you can read the second question, a particular type of cell was engineered to express different amounts of N-cadherin. So in a cell, we have different amounts of N-cadherin. Each batch in the table represents a different amount of N-cadherin on this cell type. So here we have 5 different cell types, A, B, C, D and E, and corresponding we have what is the quantity of N-cadherin in each kind of cell type. So based on these values, how much N-cadherin is expressed per cell, arrange them in the order they would sort. So if we now mix these cells, cell type of A, B, C, D, E, we mix all the type of cells. The question is, what is the order in which they would sort from outside to inside? So what cell line will occur will occupy the outer layer, and gradually as we move we reach the inner core. Based on the concentration of N-cadherin, you have to determine, if you mix all these cells, this will form a ball of cells. Now in the ball of cells, which kind of cell will occupy which layer? You just have to uh, think about it and tell me the answer in a layer based manner. So the options are, they would remain mixed as they are the same type of cells with the same type of cadherin molecule. Option B, EDBAC. EDBAC. Option C, CABDE. Option D, EBCAD. You have to remember outside to inside what is the orientation of these different kind of cells based on the concentration of N-cadherin. I think this is an easy question for you guys and you will be able to answer this. Let me know in the chat box what do you think is the correct answer. I'll wait for a minute and collect all the three answers. So Sakshi, Simontini and Ajay, you guys are the ones attending the class. I think Ajay has a question from the previous slide. External and internal path of cadherin is same or different? No Ajay, external part consists of calcium binding domains or extracellular domains. They have different function, and internal part, internal cytoplasmic part, has binding sites for beta catenin and all these linking proteins, right? So external part and internal part are not same. So the protein, you can divide a single protein into different domains, and each domain has a particular function. So the extracellular domain has calcium binding function and also homotypic interactions that I mentioned. The transmembrane domain, which is occurring inside the cytoplasm, in the membrane, transmembrane domain has a function to anchor the cadherin protein, and the intracellular domain has the function of binding beta catenin and vinculin and all this interaction and linking the cell with linking the cell cytoskeleton with the cadherin interaction. I hope it is clear. And Ajay has mentioned, yes, no, the protein has extracellular domains which can recognize calcium. So it has calcium binding pockets, as well as extracellular domain can interact with another extracellular domain of the same type of cadherin. So once it binds the calcium, then it has a conformational change, and then that domain can now recognize. So in presence of calcium, there will be strong interactions. Yeah. So Ajay has mentioned C, A, B, D, E, and Sakshi is also saying option 3. Simontini, will you be able to answer? I think this is a simple question, and if you have gone through the lectures, or even if you base your answer based on the first question, you will be able to answer it. Okay, seems like she's shy to answer. It's okay. Let me reveal the correct answer to this question, which is option C, C A B D E. So Ajay and Sakshi have both correctly answered this question. Well done. So we see here in the first explanation here, we saw when we were solving the first question that the amount of cadherin also matters. So if there are more number of cadherins or more concentration of cadherins, then we can have such homotypic interactions in more number. So here if you see the lowest number of cadherin here is C cell type with 10 counts per cell. So this guy has a 10, this cell type has 10 cadherins. So it can form 10 adherence junction, let's say, one-to-one interaction. Similarly, if you see A has 27, B has 35, D 50 and E 73. So layer C will form the weakest adherence junction, and layer E will form the strongest adherence junction. So if you measure the capacity of single adherence junction, it won't be different from A to B to C to D, because all these cells are the similar type. But what is the difference here is the number of adherence junction, or the number of interaction the cell is formed. That is why the weakest interaction will form the outermost layer, 10 n cadherin cell per count, and the E layer with 73 n cadherin count per cell will form the inner core. So let us see, if you remember this famous experiment by Steinberg and Takeichi, they have shown here that the ability of cells to sort themselves based on amount of cadherin expression. They have seen this. So in this graph you see aggregate surface tension and surface cadherins per cell. It's a direct answer to this question. So we have different kinds of cadherin, N cadherin, P cadherin and E cadherin. If you follow just the N cadherin, if you see the red dots here, along with the increase in surface cadherins per cell, we see an increase in aggregate surface tension. You see here, this point, this red point here, it's greater than 200. So if we have more than 200 N cadherins in a cell, the surface tension is also greater than 5 per centimeter square. So it has a linear relationship. Greater the amount of cadherin, greater is the surface tension. Similarly, if you see this red dot here, it has less than 50 molecules of N-cadherin. So the surface tension is also very low, 1, more than 1. Similarly for P-cadherin, if you see, there is a linear relationship again. So the more number of cadherins, the more surface tension the cell has. Now if you remember this Steinberg and Takeichi experiment, what they have done, green cells have 2.5 times more N-cadherin than red cells. Green cells sought to center with red cells at periphery. Since this interaction is very strong within the green cells, or the cells with higher cadherin, they will form the core of the cell mixture, and loose interactions will form the periphery of the cell mixture. So that is how we decided that CABDE will be the structure. So again, what it has to do with this explanation? Cells can rearrange themselves into most thermodynamically stable patterns. So always these cells will try to attain the most thermodynamically stable organization. That means cells with greater surface tension will accumulate together in the center, and cells with lesser surface tension will move to the outside. That is what I have written here. This is again from the Takeichi experiment. We have P cadherin greater than E cadherin. Here the surface tension. So the P cadherin is occupying the center of the ball, and E cadherin on the periphery. When we have surface tension of P cadherin equal to E cadherin, so we do not see any organization, cells are dispersed apart. When we have E cadherin surface tension greater than P cadherin, so E cadherin is making the core while P cadherin making the outside. So cells with higher surface tension with more NCAD expression would tend to move inside. Cells with lower surface tension with less NCAD expression would tend to move outside. That's why we have the answer. Our order is C, A, B, D, E, where C was the lowest and E was the highest NCAD expression. Is it clear to the students? It's a very simple explanation, and students have also have answered very correctly. Very good. Is it clear to the students? Just let me know in the chat box. Great, great, Sakshi. Thanks. Moving on to the next question, number 3. Which of the following is false? Let me remind you that there can be more than one statement which is the answer of this question. Which of these is false? Which of this is a false statement? Changes in actin cytoskeleton and cadherin adhesion leads to formation of extended branches from a single cell during trachea development in Drosophila. So first statement is talking about trachea development in Drosophila. Changes in actin cytoskeleton and cadherin adhesion leads to formation of extended branches. Option B, twist expression is an example of external cue regulating morphogenetic changes of cells. Here we are talking about the factor twist is an example of external cue regulating morphogenetic changes of cells. The statement 3, the first step in cell migration involves activation of proteases to cleave off existing attachment of the cells. What is the first step in cell migration? And option 4, myosin 2 migrates from basal to apical side only in the ventral region of Drosophila blastodon. So these are the 4 statements, and you guys have to pinpoint which are the false statements, which is not correct. Great, Ajay and Sakshi are very prompt in responding. They have already answered option 2 and option 3. Great. Simontini, are you comfortable with this question? Can you answer this? See guys, if you have been through the lectures, then it's a piece of cake. There is no difficulty in this. Just that these statements refer to different topics that professor has covered. The topic of trachea development in Drosophila, twist expression in the ventral patterning of the Drosophila, and all these 3 statements. The third statement is in migration, epithelial to mesenchymal transition and migration, if you guys know of that. And the fourth statement is also for ventral Drosophila. So I think we should move forward. Ajay and Sakshi have both answered very correctly again. Option 2 and option 3 are the false statements. Twist expression is an example of external cue regulating morphogenetic cells is a false statement. Twist expression is an internal cue, and the first step in cell migration involves activation of proteases to cleave off existing attachment of cell. This is also not true. The first step is paracrine signaling, which will downregulate the expression of cadherins and other junction proteins and adhesion proteins, so that the cell can get rid of these junctions or these tight placements. So let us get into little more detail. So the first statement that we read about is trachea formation in Drosophila. A brief representation of trachea formation. So here in the first stage, stage 11, in the lowest stage, we have tracheal epithelium cells which experience an secretory protein called BNL or branchless protein. some cells secrete this BNL protein. Now what this BNL protein would do is activate some cells in the tracheal epithelium called the leader cells. Leader cells are basically those cells which respond the earliest to the branchless protein, and under the influence of this branchless protein, the leader cell will now undergo shape, shape change and morphological change by rearranging its actin myosin cytoskeleton via Rho GTPase mediated process to migrate and form the secondary branches. So there is involvement of again Rho GTPase, there is actin myosin cytoskeleton changing, and during this migration, cadherin proteins are regulated such that epithelial cells can migrate over one another to form a tube while keeping their integrity as an epithelium. So cadherin proteins here, if you see between 3 and 5, the cadherin proteins, the type of cadherin proteins which is holding together, and the property of between 1 and 3 is very different, because 1 is still attached by a adherence junction to the cell number 3, but 1 also has to extend its cytoskeleton and move over other cells. So during this migration, cadherin proteins are regulated such that epithelial cells can migrate over one another to form a tube-like structure. So we see here a tube-like structure while still keeping their integrity as an epithelium. So this is trachea formation in Drosophila, and the cell shape changes are brought about by actin myosin cytoskeleton and Rho GTPase mediated dynamics. So statement 1 is not false, it is true statement. Then coming on to statement 2 and 3, which is the correct answer. Here is an uh, example of cell migration. So we have apical polarity complex adherence junctions where we have E-cadherin in epithelial cells, basal lamina, integrins. So there are some proteins which can attach the cells to the basal lamina and the extracellular matrix. There are E-cadherins which can attach cell to cell junctions and other kinds of junctions. So what happens, the first step is signaling by paracrine factor. So this cell which is undergoing migration or which is planning to undergo migration will receive some paracrine factors from nearby environment or from nearby cells, and paracrine factor from neighboring cells activate gene expression in target cells, thereby instructing the target cells to down-regulate their cadherins, release their attachment to laminin and other basal lamina components. So what these paracrine signaling factors would do, it would signal the cell, remove all your junction counterparts, remove your E-cadherins, remove your adherence junction proteins, also remove integrin proteins so that you can lose your connection with the basal lamina as well as other cells. So from a closed packed structure, a cell can be in a motile state. So that is the first step in signaling. And cleaving by proteases of the basal lamina is the last stage, which was falsely mentioned in the option 2. That's why it is false answer. And twist is an internal cue that mediates morphogenetic changes in cells. Twist is not an external cue, as we saw here in the trachea formation. Branchless is a protein which is externally influencing the tracheal epithelium cells, while twist is a protein that is encoded in the cells themselves and which causes morphogenetic changes in the cell. We will see further that in the Drosophila ventral side, how the cell shape changes is mediated by snail and twist. So is it clear to the students? Well, you guys are answering very nicely. Till now you have answered all correctly. So statement 2 and 3 were false. Twist is not an extracellular cue, and the first step in is paracrine signaling in cell migration. I hope it is clear to the students. Just let me know in the chat if it is clear, and then we'll move to the next question. So Ajay has a question in the chat I just saw now. Can we change surface tension? So in that experiment that we saw there, Takeichi and the other scientists have used different kinds of catherines. Not the type, so within the type we can actually change the surface tension. So it will depend on the number of extracellular domains a catherine has, and what is the so There can be many genes coding for extracellular domains and many variants of that. So that can affect the surface tension of a cadherin. So between N cadherin, there might be, if let's say if an N cadherin has 4 extracellular domains and the other one has 6 extracellular domains, then surface tension between them will differ. The 6 extracellular domain guy will be stronger, will be having higher surface tension. So such isoforms exist because genes code for all these proteins, and these can undergo alternative splicing to give different kinds of cadherins. So they are N-cadherin categorized because they are expressed in the neuroepithelium, E for epithelial, but within them they can have differences. So N-cadherin is a family of cadherins, so we can change surface tension. Practically, yes. Okay, cool. Yeah. Hmm. Okay. Yeah, yeah, yeah. Yes. Yeah, definitely. Okay. So the fourth question, arrange the following in order in which they occur when mesoderm forms during gastrulation in Drosophila. So here we have uh, the mesoderm formation in Drosophila, where we'll study about snail twist. So we already have some statements. So you have to arrange these statements in order of their occurrence when mesoderm forms during gastrulation in Drosophila. Statement 1, change in cadherins and actin cytoskeleton. Statement 2, twist and snail expression occur in the cells of the ventral region only. Statement 3, migration of rho and beta catenin. Statement 4, apical constriction. Now, which of these order of the statement do you think is the correct option? Option A, 3214. Option B, 2143. Option C, 2314. Option D, 1423. Option E, 4132. Guys, take a minute and try to answer the question. The question is before you in the slides. Take a good read at the statements and just let me know in the chat box what do you think is the correct order of events that occur during mesoderm formation in Drosophila. Oh, Sakshi and Ajay have already answered. Sakshi is saying 3. 3 means I guess option 3, and Ajay is also answering option 3. 2314 is what he was saying. I think the way you guys are answering today will end our class very soon because you guys are so fast. By the time I finish reading the question, you guys are ready with the answers. Very good, very good. It's good to see that. So let's not wait any further. Let me display the correct answer. The correct answer is 2314. Ajay and Sakshi are very correct, and they have answered very correctly. So if we look at the mesoderm induction in Drosophila, the mesoderm induction happens on the ventral side of the Drosophila. Migration is started by the expression of twist and snail. So these are two factors that we encountered in the previous question. Twist. So here is a representation cross section of Drosophila embryo. This side represents the dorsal side and this side is the ventral side. What happens, you see these red dots are twist expression domain and the blue is a snail expression. So twist and snail only express in the ventral side of the Drosophila, and due to their expression, what happens is myosin 2 is a protein which lines the basal side of the epithelium. If you see these dark blue proteins here line the basal side, and this outer layer is the apical side. So myosin 2 generally lines the basal side of the protein, forming lamina, basal side of the lumen, forming an impenetrable layer. So basal membrane, nothing can penetrate it. Means the cell cannot. It's a hard surface. So what this expression of twist and snail does, it flips the expression of myosin 2 from the basal side and brings it to the apical side. So here if you see in the apical side, the blue expression is coming up. That means myosin 2 is now expressing for those regions expressing twist and snail at the apical side. Thereby these cells now have a opening in the basal membrane through which they can migrate. So hence this invagination comes up, migration is categorized. This will then form induce the mesoderm. This will induce the mesoderm in the Drosophila. Rho buildup on ECAD causes actin polymerization and migration. So this is the expression of another two players, Rho GTPases and beta catenin, which forms the adherence junction and actin cytoskeleton linkage. So here we see that Rho and beta catenin were present in the basal side, and only beta catenin was present on the apical side of some cells, because apically we have adherence junction. But when these expression of twist and snail occurs in the domain of myosin 2 expression, we also have Rho and beta catenin expression, and Rho expression is lost from the basal side, and it couples with the beta catenin, and then this migration can happen with the apical side having Rho and beta catenin expression. So thereby now we can decide the order of the events that are happening. So the first order would be twist and snail expression occur in the cells of ventral region only. Then migration of Rho and beta catenin along with myosin 2. Then we have change in cadherins and actin cytoskeleton. So migration of Rho and beta catenin will influence actin polymerization by Rho and beta catenin, change in cadherins, and apical constriction where the invagination can proceed. So this is a brief about ventral mesoderm induction in Drosophila, how the expression of certain factors like twist and snail can influence uh, the change in cell cell adhesion and cytoskeletal dynamics to induce migration. I hope this is clear to the students, and I'm happy to see that students are answering very correctly. Good. Amoeba. Amoeba is a single celled organism. So yeah, yeah. So the protrusion is there because amoeba also has cytoskeleton like actin filaments. It also forms the pseudopodia. So those are the end feet or leg-like structures through which the actin polymerization is occurring, which is pushing the cell membrane of amoeba and then forming a leg-like structure. But developmental biology is study in which all these individual processes like cytoskeletal arrangement, like some signaling, is processing the development of an organism or some organ. So that won't be applicable in amoeba, but the individual phenomenon will be seen in amoeba, like cytoskeletal rearrangement. That is the exact way amoeba moves by forming lamellopodia, which is called pseudopodia in amoeba. So you are absolutely correct, Ajay. Yeah. Yes. Okay. That's a unique line of thinking. Yeah. Always ideas are always welcome. You should think. It's really good. But yeah, we will read more about it. Now let's move on with the fifth question that we have today. In the developing ectoderm, which of the following is true if epidermal cells were to made express N-cadherin? So this is the so all these topics have been covered in the lectures, and if you read the book, reference book, Scott Gilbert, you'll find them very beautifully explained, and Professor Subramaniam has also explained it very nicely. If you have followed through that, you'll be able to answer this question in a jiffy. So in a developing ectoderm, which of the following is true if epidermal cells were to made to express N-cadherin? Options are: cells would normally sort to form neural tube and epidermis. Option B, the boundary between neural tube and epidermis will not form. Option C, the boundary formed would be similar to what would happen if N-cadherin was knocked out in the presumptive neural tube cells. Option D, epidermal cells will also form the neural tube. Option E, multiple neural tube structures will form. So students, you have to tell me, which options do you think is true in case of ectoderm development and cadherin molecules? Wow, Ajay and Sakshi have already answered again. Option 2, 3, good. So option 2 and 3, the boundary between neural tube and epidermis will not form. Boundary formed would be similar to what would happen if N-cadherin was knocked out in the presumptive neural tube cells. So I'm happy to announce that all the options are correct, exactly correct. Great. So option 2 and 3 are the true answers, or is the true statement. What would happen if epidermal cells were expressing N-cadherin? So basic knowledge about cadherin expression, very basic. Epidermal cells will express epidermal cadherin, which is E-cadherin. As we see here, this is a cross section of a neural tube before closure. These are the epidermal cells expressing epidermal cadherin. And the neural cells, the cells which will give rise to all neural structures, will express N-cadherin. E-cadherin epidermal, N-cadherin neural. So what would happen here? In the developing ectoderm, the expression of N-cadherin is important in separating precursor of neural cells from the precursor of epidermal cells. So if you know this fact, that all the neural structures arise from the ectoderm itself. So the ectoderm is basically expressing E-cadherin. But when the neural tube will start forming, or the neural patterning will start, all those cells which are going to give rise to neural structures will express N-cadherin. So that's how there will be a differentiation between the epidermal structures and the neural structures. So what would happen if we forcefully express, sorry, if forcefully E-cadherin cells were made to express N-cadherin? So what would happen? Initially, all embryonic cells contain E-cadherin, but those destined to become the neural tube lose E-cadherin and gain N-cadherin. If epidermal cells are experimentally made to express N-cadherin, or if N-cadherin synthesis is blocked, which was also one of the statements, in prospective neural cells, the border between skin and nervous system fails to form properly. So this border, this distinction that we see here, that this part is going to be neural, while this part is going to the epidermis. Now if we express neural cadherin in the epidermis, then it will also behave as a neural cell or a neural precursor cell, and there will be no boundary between the neural tube and the skin. So what would happen then? The border between the skin and the nervous system fails to form properly. That will happen. So here we have a wild type image here and N-cadherin knockout image. And we see here N-cadherin knockout in the neural cells. So here we see if epidermal cells were to express N-cadherin exogenously, which they should not, because they should be expressing E-cadherin, the boundary between neural tube and epidermis will not form, and the boundary form would be similar to what would happen if N-cadherin was knocked out in presumptive neural tube cells. So now if N-cadherin is knocked out in the neural tube cells, then also the boundary will not form, because the boundary will only form when there is epidermal cells expressing E-cadherin and neural cells expressing N-cadherin. Now if neural cells have N-cadherin knocked down, then it will also behave similarly, There will be no distinction between the epidermis, the skin, and the nervous system. I think you guys have answered it very correctly, and I hope the explanation is clear. Quickly let me know in the chat box, and we will move ahead to the sixth question. Great, great, great. We are moving very fast today, all thanks to your correct answers. So the sixth question is a match the following question, right? We have factors, FGF proteins on the left, and pathways they affect in the right. FGF1, limb development; FGF7, blood vessel formation; FGF8, skin development; FGF2, regeneration. This is a completely memory based question. I won't, you can guess the answer, or if you know it very well, very good, well and good. But if you do not, it's okay. It's a memory based question. Sometimes you remember it or not. But there are different FGF factors here, and which phenomena do they target? You have to identify. So options are given on the screen: 1B, 2C, 3A, 4D, 1A, 2D, 3C, 4B, 1C, 2B, 3D, 4A, 1D, 2C, 3A, 4B, 1B, 2A, 3C, and 4D. Now, out of these, which do you think is the correct matching? Okay, Sakshi has answered option 4, which is 1D, 2C, 3A, and 4B. Okay, let's see. DCAB is what Sakshi is answering. Ajay and Simontini, will you be able to answer? You can just guess what you think is the correct answer. It's okay if your answer is not correct. We are anyway going to discuss about the answers and the concept behind it. So do not feel shy. I'll wait a minute for you guys to answer. Okay, Ajay is answering option 1, which is BCAD. Simontini, will you be able to answer this? Okay, okay. Okay, it's fine, it's fine. We'll move on to the correct answer of this question. So the correct answer, Sakshi gave the correct answer. DCAB is the correct sequence. FGF1 for regeneration, FGF2 for FGF7 for skin development, FGF8 for limb development, and FGF2 for blood vessel formation. So this is the correct order. It's okay if your answer is wrong, Ajay. You do not have to worry about it. So let's have a brief lookout of FGF pathway. FGF stands for fibroblast growth factor, and this is how the pathway is mediated. The ligand of FGF can be of these different types: FGF1, 7, 8, 2. So there are different ligands, and it signals through receptor tyrosine kinases. So these are the receptor tyrosine kinases. When two FGF when FGF ligand binds, it homodimerizes the RTK or receptor tyrosine kinases, which can then autophosphorylate, and then GF, RAS, RAF, MEK, ERK pathway will follow. So these phosphorylation on these receptor tyrosine kinases can harbor adapter proteins like SOS, which can then activate GEF, which is a guanine nucleotide exchanging factor. GEF protein and GAP protein act in tandem to change the state of another important protein called RAS. So RAS is a protein, RAS is a GTPase. When RAS is bound with GDP, It is inactive, but when the GDP is exchanged with GTP, RAS is active. So GEF and GAP play a role in cycling the RAS between GDP bound and GTP bound states. Now when the RAS is in GTP bound state, it can activate another protein called RAF, and RAF can activate another protein called MEC. RAF can phosphorylate and activate MEC. MEC can phosphorylate and activate ERK. Now ERK can phosphorylate a transcription factor which was inactive in the nucleus. Phosphorylated transcription factor will be active, and then this transcription factor can sit down on different FGF responsive elements and turn on gene transcription. So this is how a schematic of FGF signaling looks like. Now coming on to the different matches that we saw in the match the following. The fibroblast growth factor family of paracrine factors comprises nearly 2 dozen structurally related members, and the FGF genes can generate hundreds of protein isoforms by varying their RNA splicing or initiation codons in different tissue. So we have different kinds of FGF due to alternative splicing. FGF1 protein, also known as acidic FGF, appears to be important during regeneration. That is what we saw. FGF1 for regeneration. FGF2 is sometimes called a basic FGF, very important in blood vessel formation. FGF7 sometimes goes by the name of keratinocyte, critical in skin development, and FGF8 for bone and limb development. Also plays a role in lens induction. So Professor Subramaniam has covered the role of FGF8 in lens induction, and FGF8 also plays an important role in limb development, while other FGF1 plays a role in regeneration, 2 in blood vessel formation, and 7 in skin development. So this is the correct order of protein affecting the function. It's okay if your answer is incorrect. This is a memory based question. Sometimes you might get such questions in the exam, but it's okay. You just have to go through your lectures once more before the exam and you'll be able to do it. I hope the explanation is clear. Just let me know in the chat with a yes or no. Then we can move to the next question. Great, great Sakshi. Thanks Ajay. So the next question, number 7, which of the following is true? Again, we have a true false statement. Option A, introducing an FGF8 releasing bead in the place of the optic vesicle will induce lens placode formation in the head ectoderm. So if we have a FGF8 releasing bead in the place of optic vesicle, will it be able to induce lens placode formation in the head ectoderm? Option B, during development, intermediate structures do not have any functional role. Option B. Option C, a particular signaling pathway can play different roles in different organisms in different stages of development. And option D, the differential hypoadhesion hypothesis states that cells interact to form a structure with greatest interfacial energy. Now out of these 4 statements, which of these statements are true? Read the question carefully and then let me know which are the true statements. One question is about FGF8 and eye induction, optic lens placode induction. During development, do we need intermediate structures or not? If a particular signaling pathway can play different roles in different organisms, and what is the differential adhesion hypothesis, if you remember it. Okay, Sakshi and Ajay have answered all except 2. Okay. So let us see what is the correct answer. Option 1 is correct. If you introduce an FGF releasing bead in the place of optic vesicle, it will induce lens placode formation. Option 3 is correct. Particular signaling pathway can play different roles in different organisms in different stages of development. and these two statements are correct. I think both of you have mentioned differential adhesion hypothesis states that cells interact to form a structure with greatest interfacial energy. So maybe we'll clear that out. Interfacial energy. Actually, you guys might be in a sense correct because it seems very counterintuitive, but if you stick to the definition or the hypothesis laid down, then I think this will not be the true statement. What let us just discuss one by one. FGF8 containing beads are placed adjacent to head ectoderm. This ectopic FGF8 will induce this ectoderm to produce ectopic lenses, express the lens associated transcription factor LMAF. So here we see contact with the optic vesicle. This is the natural one. While contact with FGF8 bead without the optic vesicle, so in a place lower to that, even if we do not have the optic vesicle, we can just place the FGF8 bead and can induce a lens placode, because FGF8 is the molecule that is secreted by the optic vesicle, which leads to induction of transcription factor LMAF, which is a lens placode specific transcription factor, can guide the formation of lens. So statement 1 is true. Then statement 3, where a same signaling pathway can have affect different structures in different organism at different developmental time. We can see that Wnt signaling was discovered in Drosophila, is responsible for Drosophila wing development, while in humans it is responsible for neural development, for cell proliferation and many others. So Wnt is a major signaling pathway. Now we have a perfect example of Wnt signaling active in two different organisms in different developmental time, affecting different functions in human central nervous system development and Drosophila wing development. Now for the fourth statement, where your and my answers do not match, differential adhesion hypothesis, what does it say? According to this hypothesis, cells congregate near other similar cells with similar adhesion molecules to maximize the bonding adhesion strength between cells by creating a more thermodynamically stable, mind the spelling mistake, thermodynamically stable, less energy costing tissue or structure. So here the differential adhesion hypothesis talks about thermodynamically stable structure, while here we had greatest interfacial energy. So interfacial energy is energy generated during surface tension, which is also true, but which can be referred to as an understanding of the hypothesis. But the actual hypothesis, what it states that is whatever is most thermodynamically stable, which is also in this case greatest interfacial energy, will form will interact and form a structure. Yes, Ajay, what were you saying? Yes, 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 Ajay. Yeah. I understood, but in such tricky places, you just go by the grammar and vocabulary. So this is not something. These answers, as you know, is not something I came up. So these answers are already there. So this is a previous assignment. So when you see differential addition hypothesis, this is an established hypothesis, and whatever it states, you have to state it. So your argument is basically correct. In an organ, we have different kinds of tissue, and those tissues will only form an organ when they are in thermodynamically stable state. Now in the tissues we can have based on surface tension, we can have based on interfacial energy, we can have also based on entropy. So we, in a system when there is less entropy, we have a stable system, right? So anything can be possible, but here the exact statement of hypothesis would be thermodynamically stable. You are absolutely correct, but if you encounter such question, please answer uh, that the whatever the hypothesis is exactly stating, right? I think this, yeah, I think this is clear to all the students. Then we will move on to the next question. Moving quite fast. This is very good. Consider a situation, question number 8. Consider a situation where thigh mesenchyme taken from a bird of a species A is recombined with wing epithelium from a bird of species B, right? A thigh epithelium from one species, wing epithelium from another species. What would happen in the above scenario? Option A, the epithelium will produce thigh specific feathers of species B. Option B, epithelium will produce thigh specific feathers of species A. Option C, the epithelium will produce wing specific feathers of species A. Option D, epithelium will produce wing specific feathers of species B. Now all these four statements can be quite confusing, but trust your logic and your science. Try to read the statement again, think about it, and let me know the correct answer in the chat. Ajay and Sakshi are answering option 2. So you have to read the question again. Thigh mesenchyme taken from bird species of A is recombined with wing epithelium from a bird of species B. Right? And then you guys are answering option 2. Epithelium will produce thigh specific feathers of species A. Okay, let us see what is the correct answer since you all have answered. Option 1 is the correct answer. The epithelium will produce thigh specific feathers of species B. So option 2 is not the correct answer, but option 1 is the correct answer, where we have thigh specific feathers of species B. So let us understand why this is the correct answer. So this is the classical wing epithelium and the mesenchyme, surrounding mesenchyme. So if we have source of dermal mesenchyme is wing, then in the wing dermal epithelium it will produce a wing feather. If the source of dermal mesenchyme is thigh, then wing epithelium will produce a thigh feather. And if the source of dermal mesenchyme is foot, then wing epidermal epithelium will produce scales and claws. Now you imagine you fit the question in this situation. All these mesenchyme, these pink tissue, are from species A. The mesenchyme taken from bird of species A. So bird of species A has these pink tissue, and this blue tissue, wing epidermal epithelium, is from bird B. This is from the species B, as mentioned in the question. So now, where did we fall back? Where did we do the mistake? Now you see, the source of signals or the source of induction signals will be the mesenchyme. Right? You agree with me? So species A bird will give the signal to the wing epidermal epithelium because it is a thigh mesenchyme. It will tell the wing epidermal epithelium, grow a thigh feather. That's it. That's the signal that species A mesenchyme is going to give. But what species B epidermal epithelium is going to do, it is going to receive the signal but then produce a species B kind of thigh feather, because in the signal it is not encoded, species specificity is not encoded. Signal is just an induction signal to form a thigh feather. Now the thigh feather will be of what origin? It would not be of A origin, it would not be of the mesenchyme origin, but from the wing epidermal origin, because the epidermis is from the species B. I hope you are understanding the subtle difference. You guys were really correct. It will form a thigh feather. and the induction signal is coming from thigh mesenchyme with species A. But the feather that will form from this tissue, na. So this tissue will form a type B thigh feather. So here again, simplifying it for you guys to understand, mesoderm is of species A is the inducer. It will induce the formation of whatever underlying tissue it is. It will produce factors which can signal the underlying ectoderm to produce certain type of structure. Let's say a wing, thigh feather, or a claw. While ectoderm of species B is the competent tissue. So we have an induction tissue and a competent tissue. It will produce structures in response to inductive signal received from the mesoderm. So whatever structure will be seen will be produced from the species B ectoderm. Therefore, we will form thigh feathers with form of species B. That's why option A will be the correct answer. So we'll have feather, but of species B, because the tissue that is forming the feather is actually the epidermis of species B, while the species A tissue is signaling the epidermis to become a thigh feather. Did you guys understand the explanation? Yes. No, no, no. Ajay, you seem to miss my point. Yes, yes, yes. Signaling is from A, right? Because, yeah, yeah. But here we are talking about different species, right? Do you understand now the point? So whatever structure, see the signal. Yeah. Hmm. No, you might be wrong here, because the signal that is coming from the mesenchyme will induce production of thigh specific feathers or whatever, but it will form the species B specific feather because it is the wing epithelium. The epithelium has a characteristic of species B. That's why it will produce a species B type of feather. It might be yellow color, red color, but if the B species is producing red color, then it will have a red feather. Controller is the mesenchyme. Okay, maybe you go back and watch it again. So if you are not able to understand, yes, yes, it can be, but if you're yeah. Okay, let me give you another example to make it clear. So let's say the genetic makeup of these birds are very different. Let's say mesenchyme from A is an ostrich and B is a parrot. Now the genetic makeup is very different, right? So whatever the signal it is the mesenchyme is giving, that might not be present in the next species. Ostrich kind of feathers. See, ostrich kind of feathers might not be there in parrot, which is actually true. So even if you keep an ostrich inducer in parrot species, then it will produce a parrot specific thigh feather. So the mesenchyme, what information it is passing is, it is passing the characteristic of the feather, that it is thigh, wing or claw. That is the common ground, but it will be species specific, because the eventually the feather that is arising from the tissue, that tissue is the bird species B. So that tissue will be of parrot origin, it won't be of ostrich origin, right? Because ostrich is just inducing, parrot will be producing it. So it might guide the, it will tell the parrot epidermis to make a yellow feather. The parrot might not have genes for it. Parrot will produce still a green feather, but it will produce a thigh specific feather, since the mesenchyme is now a thigh mesenchyme. I hope this is helping you to understand, or you can read it up yourself. Yeah, yeah. If you give legs, it will produce. It was, yeah. Yes, but you see those. Yeah, so those signals are from the same species, right? The species is same. It is in the chicken or in the human, or you just provide the protein. So the genetic makeup is same. I think here the two species are mentioned here, A and B, because the genetic makeup is different. So even the inducing, yeah, you can go back and clear your doubts, and you can ask me in the next class. We can talk about this, but let's move on to the next question. What property of induction is this an example of? In this case, this is the induction problem that we faced in the earlier thing, the wing thigh mesenchyme with the word epithelium. So what property of induction is this an example of? Option A, regional specificity of induction. Option B, genetic specificity of induction. Option C, permissive induction. Option D, reciprocal induction. Option F, none of the above. What property of induction did we see just now? Thigh mesenchyme and the epidermis from the bird, wing epidermis. Ajay is answering option 4, combination of regional and genetic specificity. Okay. Sakshi and Simontini. See, we are reaching towards the end of these problem session. Question number 9 and 10, and we are done. So I would encourage all of you guys to answer. Simontini and Sakshi. Sakshi has been answering. Do not feel shy. This is actually a session conducted to have discussions, just like we earlier used to have, so that you can clear your doubts, right? So, okay, Sakshi is saying don't know. It's perfectly fine. And Ajay has answered very correctly. Combination of regional and genetic specificity of induction. This is the property uh, that we encountered, right? So let us have the definitions of these kind of all these kinds of inductions. Permissive induction occurs where the responding cell is already committed to a certain fate and requires the inducing signal to proceed in the developmental pathway. So this is not the case, since responding cell is the wing epithelium. Wing epithelium is still not committed to a fate. Wing epithelium can form the forelimb feathers, the thigh feathers, and the claw. Reciprocal induction refers to mutual process in which two or more interacting cells or tissues influence each other's development, differentiation or function. Reciprocal induction, both the participating tissue will induce each other. Regional specificity of induction refers to the phenomenon in which fate of group of cells or tissues is determined by signals from neighboring cells in a spatially specific manner. So in a spatially specific manner, from the forelimb, for the thigh or from the foot can induce different. And genetic specificity of induction is what I was referring and discussing with Ajay, that genetic specificity will be preserved refers to highly controlled specific interaction between cells and tissue during embryonic development. This specificity ensures that appropriate signals are exchanged between neighboring cells, leading to precise differentiation of various types of cell and formation of distinct structure and organs. So genetic specificity. So here you might we might come back to the earlier discussion that we were having. So the genetic specificity will be of species B because the structures, the cells and tissues that are making up the thigh feather is from the species B. Whatever signal they received is from the species A, but the cells and tissue which are going to act in order to form the structure are from species B. That's why they will form a thigh feather of species B. Ensures genetic specificity. Yes, Ajay. Oh no. Yeah, but the main thing is if you are able to clear your doubt, then it would be a good thing. So if you understand, the induction signal is something, a molecule or a factor that is getting secreted from the mesenchyme, and the epithelium is responding to it, right? That molecule or something. Now if you see, that molecule can be a ligand, can be a something, but the ligand will induce signaling with respect to the acting species, right? Or the genetic makeup of the cell, the kind of cell that is forming the tissue. I mean, you can you understand? The signal will come, but the function that will be performed or the final product that we get will be the acting tissue from where it is originating. Yeah, yeah, good. So it's good that we could clear this discussion here with this question. And moving on to the last question, uh, we answer this and we are done with the practice problem session. In the developing mammalian kidney, the metanephrogenic mesenchyme stimulates ureteric buds elongation and branching. So this is a paragraph from nephrogenesis. The ureteric bud branches into the mesenchyme to form aggregates, which then differentiate into renal nephron. This is an example of dashed in cases where each tissue is cultured individually without the other dash. So there are two fill in the blanks. You have to fill in the blanks with the correct options. Option A, reciprocal induction, and number 2 will be both will not form functional structures. Option B, regional specificity of induction, both will not form functional structures. Option C, genetic specificity of induction, ureteric bud will form branches but the renal nephron will not develop. Option D, reciprocal induction, both will form respective functional structures. And last option, option E, genetic specificity of induction, the ureteric bud will not form branches but the renal nephron will develop. Now, which of these do you think is the correct answer for filling in the blanks in the paragraph? This is about nephrogenesis, generation of nephrons, development of nephrons in the kidney. Take a minute to read the sentences and fill in the blanks. Okay, Sakshi is answering maybe 3, genetic specificity of induction, and Ajay is answering 4, reciprocal induction, both will form respective functional structures. So I'll urge you guys to read the paragraph carefully and then decide your answers. What is the general phenomenon of nephrogenesis known as, is an example of what kind of induction, and in case when each tissue is cultured individually without the other, what will happen? Okay, let me reveal the correct answer. The correct answer is option A. Option A is the correct answer. Nephrogenesis is an example of reciprocal induction, and when both the mesenchyme as well as the ureteric bud are cultured individually without the other, then they will not form functional structures because there is no reciprocal induction when they are not together. They are cultured individually, they will not form any structures, and this is an example of reciprocal induction. So let's learn a bit about nephrogenesis and reciprocal induction. So the metanephric mesenchyme is the underlying mesenchyme. It will secrete some factors like glial-derived neurotropic factors signaling to the ureteric bud. Ureteric bud is the underlying tissue. So once the metanephric mesenchyme signals the ureteric bud, GDNF, glial-derived neurotropic factor, binds to receptors inducing branching and outgrowth of ureteric bud. So signal from mesenchyme induces branching and growth of ureteric bud, and then now that the ureteric bud has grown, outgrowth of ureteric bud into the mesenchyme induces mesenchymal to epithelial transformation. So these ureteric branches, when penetrate into the mesenchyme, they will now cause epithelial mesenchyme to epithelial transformation. So the underlying mesenchyme will now form epithelium. Inductive signals from UB or ureteric bud promotes nephrogenesis characterized by differentiation of mesenchyme into epithelium, in which further cell growth and configurational changes evolve into nephrons. So this is a classic example of reciprocal induction. First, the mesenchyme signals the ureteric bud and causes its outgrowth. Then the ureteric bud also signals back to the mesenchyme, causing the mesenchyme to undergo an epithelial transformation and form the nephron. So these both the tissues are required for the correct formation of nephron. If one of the tissues is missing, then there won't be any reciprocal induction and correct nephron functionality will be lost. So the answer is option A, reciprocal induction. And when each tissue, like each tissue means mesenchyme and the ureteric bud, are cultured individually without the other, then they will not form functional structures, which is the nephron. They will not form it. I hope it is clear to the students. So sometimes questions can be very tricky. You guys have to go through it, read it carefully, and then decide your answers because options will have everything. Options will always convince you to tick the correct, tick the wrong option. Wrong options look more convincing. Let me say you. Yes, Ajay, you have some question. Yeah, means here it means yeah, mesenchyme and the ureteric bud are different. Means they are separate. Then how will it affect the nephron formation? Yeah, yeah, yeah. Yeah, that's why I was saying that wrong options will look very tempting to click, but you must reassure yourself, read through the question nicely, take some time, and then decide on your answer based on your concepts, and you will always be successful. With this, we have come to the end of the problem solving session. We are already finishing half an hour early. It's very good because all the students were answering very promptly, and I hope it was a fruitful session and the discussion could give some insights into the course. I'll stop presenting my screen now. So this is how I was also expecting the discussion to be. and I hope the students have learned something, and discussion was very nice. So keep following your lectures and refer to the TA sessions regularly. So you also have another TA session by Parth on Mondays. You can attend that. You should attend that. And I would encourage you, if you have any friends, you can tell them if you find any benefit in the TA sessions, they can also join. And yes, okay. But you see, because we we are PhD students, and yeah, because yeah, this time is actually fixed. So when we registered for NPTL courses, NPTL was asking us whenever we are free. So I selected personally this Saturday time slot from my schedule. So Sundays now it cannot change actually. So this is the permanent time. It will be very difficult to change the slot. I appreciate it, people who are joining on Saturdays and on Mondays, and that's why we have this recordings and slide sessions where I'll upload the slides and the recordings. So anyone who misses the class can still refer to the problem solving sessions. I think we have to go with this similar format. Yeah. So that depends on the students how, yeah, how interested the student is will also matter. See, since I am still having four, five few students attending my live interaction class on Saturday, I know these students are quite motivated and really want to um, learn developmental biology. It's fine. So the student also need to be motivated. But I'm sorry, the class timings could not change. Saturday evening would be the class timing. But the recordings will be available. You can refer to them multiple times whenever you want, whichever question you feel like. And if you have any doubts, save it for me for the next class while we solve practice session week 6. And again, I'll upload this in YouTube. And it's good to finish early today because students were answering very well. And I encourage such participation in the future classes. I'll end this call, and I hope you could learn something today. We had lots of fun. Bye bye. Thank you. Thank you.